Let's go back to the basics and explore the technology platform that has been regarded as the gold standard for many years. Yeah, you guessed it, I'm talking about Sanger sequencing by capillary electrophoresis. Many might ask, why is it called Sanger sequencing? Well, Sanger sequencing is named after the inventor of this groundbreaking technology, Dr. Frederick Sanger, who I like to call Fred, who developed this method over 40 years ago in the mid-70s. So what are the basics of Sanger sequencing? Let's take a look at our lab book. It all starts by having a short primer binding next to the region of interest. In the presence of the 4 nucleotides, the polymerase will extend the primer by adding on the complementary nucleotide from the template DNA strand. To find the exact composition of the DNA sequence, we need to bring this reaction to a defined stop that allows us to identify the base at the very end of this particular DNA fragment. Sanger did this by removing an oxygen atom from the ribonucleotide. Such a nucleotide is called a dideoxide nucleotide. This is analogous of throwing a wrench into a gear. The polymerase enzyme can no longer add normal nucleotides into this DNA chain. The extension is stopped, and we now need to identify what it is. We identify the chain terminating nucleotide by a specific fluorescent dye, 4 specific colors to be exact. Sanger sequencing results in the formation of extension products of various lengths terminated with dideoxide nucleotides at the 3' end. The extension products are then separated by capillary electrophoresis, or CE. The molecules are injected by an electrical current into a long glass capillary filled with a gel polymer. And during CE, an electrical field is applied so that the negatively charged DNA fragments move toward the positive electrode. The speed at which the DNA fragment migrates through the medium is inversely proportional to its molecular weight. This process can separate the extension products by size at a resolution of 1 base. A laser then excites the dye-labeled DNA fragments as they pass through a tiny window at the end of the capillary. And the excited dye emits a light at a characteristic wavelength that is detected by a light sensor. Software can then interpret the detected signal and translate it into a base call. When the sequencing reaction is performed, in the presence of all 4 terminated nucleotides, you eventually get a pool of DNA fragments that are measured and separated base by base. What you will get in the end is a data file showing the sequence of the DNA in a colorful electropherogram, and a text file which you can use to answer the questions you may be asking. And that, in a nutshell, is Sanger sequencing. For more information, download the free DNA sequencing guide through the link below. I hope this video was helpful on the basics of Sanger sequencing, and I'm sure you'll have more questions. So submit your questions at thermofisher.com/ask, and subscribe to our channel to see more videos like this. And remember, when in doubt, just seek it out.
Pyrosequencing technology capitalizes on elegant chemistry to deliver real-time quantitative sequence data. 4 enzymes convert the synthesis of a DNA molecule into a pattern of light signals. DNA elongation catalyzed by DNA polymerase is accompanied by the release of pyrophosphate. ATP sulfurylase converts pyrophosphate to ATP. ATP powers oxidation of luciferin by luciferase, which generates a light signal recorded as a pyrogram peak. Finally, a pyrase removes any unincorporated nucleotides remaining in the reaction. The light signal is proportional to the amount of nucleotides incorporated into the synthesized DNA strand. When 3 nucleotides are incorporated by polymerase, 3 molecules of pyrophosphate are generated. The 3 pyrophosphate molecules are converted to 3 ATP molecules, and these power an equimolar oxidation of luciferin. The result is a recorded triple peak. The remaining unincorporated nucleotides are removed from the reaction in preparation for the next dispensation of nucleotides. Nucleotides are added to the reaction in a predefined dispensation order. If during a dispensation no nucleotides are incorporated, no pyrophosphate is generated, and consequently no light signal is recorded. These blank nucleotide dispensations resulting in no signal can be used as built-in negative sequencing controls. Another dispensation of nucleotides takes place. Pyrophosphate is generated, then converted into ATP, and a corresponding light signal results from luciferin oxidation, in this case, a double peak. In this manner, the pattern of light signals recorded in a pyrogram is an easily interpreted representation of the true nucleotide sequence of the analyzed DNA.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub.
Discover the possibilities of next-generation sequencing.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is the gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2, each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera which records the appropriate sequences of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other two methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina high-seq machines. The iron proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and much more. To be able to access our services, please visit our website at www.abmgood.com, and from there, click on the NGS Sequencing Services link. This will load our NGS service webpage, which details all of our available services. Clicking on a service of interest will showcase the technical details, pricing, and bioinformatics solutions that are related to that particular service.
Good grammar and spelling are important, but if you want to write essays that inspire, messages.
Please leave your questions and comments below, and we will answer them as soon as possible. For more information, please visit our knowledge base at the link provided below. Thank you for watching.
Planning a sequencing experiment? I'm sure you have a lot of questions, like which is the right sequencing method for your application? Sanger or next generation sequencing? Let's take a look at both in this episode of Seek It Out. Before the 1990s, sequencing was done the old-fashioned way. using gels and manually calling bases. Then, Sanger sequencing by capillary electrophoresis was introduced. This let labs sequence long DNA fragments with automated base calls. As the technology evolved, next-generation sequencing, or NGS, transformed the way that sequencing was performed. Let's take a look at our lab book. NGS is similar to Sanger sequencing in that you sequence DNA fragments, but an NGS is massively parallel, and this allows millions of fragments to be sequenced in a single run versus Sanger sequencing, which only produces one forward and reverse read. Researchers today have many choices when deciding which sequencing technology to use for their clinical research. Sanger sequencing with 99.99% accuracy is the gold standard for clinical research sequencing. However, newer NGS technologies are becoming common in clinical research labs due to their higher throughput capabilities and lower cost per sample. But which one should you use, Sanger or NGS? Here's what you have to consider. With its ability for long reads and gold standard accuracy, Sanger sequencing is perfect for sequencing single genes, sequencing 1 to 100 amplicon targets at the lowest cost, sequencing up to 96 samples at a time without barcoding, microbial identification, fragment analysis, high-throughput genotyping using, for example, Snapshot, microsatellite or STR analysis, and NGS confirmation. Next-generation sequencing offers fast turnaround time and takes only about 4 hours to complete a run. So NGS is perfect for interrogating greater than 100 genes at a time cost-effectively, finding novel variants by expanding the number of targets sequenced in a single run, sequencing samples that have low input amounts for starting material using, for example, ion AmpliSeq library preparation, which requires as little as 10 nanograms of input DNA, sequencing microbial genomes for pathogen subtyping to enable research of critical outbreak situations. With Applied Biosystems' gold standard for Sanger sequencing and simple, scalable ion torrent next-generation sequencing, Thermo Fisher Scientific has the right sequencing platform for every research application. I hope this video was helpful on finding the right sequencing tool for your application, and I'm sure you'll have more questions, so submit your questions at thermofisher.com/ask, and subscribe to our channel to see more videos just like this. And remember, when in doubt, just seek it out.